Okay, so good morning once again. And as you can see on my screen, we are looking at osmosis today. Now, in our last class, just as a recap, in our last class, we introduced the topic cell physiology, and I was able to outline the objectives of the topic. That means some of the things that you are expected to know or to have understood by the end of this topic. So I was able to outline the objectives of the topic. I also went ahead and outlined the objectives of our last class. And as usual, I'd also like to outline the objectives of our class today. But before that, I just want us to remind ourselves of what we learned in our last class. And without wasting time, in our last class, we introduced the topic cell physiology, and we were able to define what cell physiology is. Apart from that, we went ahead and looked at some of the physiological processes that take place some of the physiological processes that take place in living organisms. We also went ahead and looked at the structure and functions, structure, properties, and functions of a cell membrane. We went ahead and looked at now the physiological processes. We were able to outline them, and we said we have got 5 physiological processes, but then at our level, we will only be looking at 3, and that is diffusion, osmosis, and active transport. Now, for the case of phagocytosis and pinocytosis, we are not looking at them at this level. But for those of you, like I said last time, who would be interested in studying biology at higher levels, you'll get to interact with those 2 other physiological processes. But again, I said as a good student, it is important for you to always do more than you're expected to do. Go ahead of the teacher, look at them, and you'll find them very interesting to know. So if you didn't look at them, you still have the time. You can look at them at your own time. Google, go to YouTube and find more about them. There are very many good videos outside there which will help you understand them. So you can do that at your free time today. So in our class today, okay, under diffusion in our last class, we looked at the definition of diffusion. How is it applicable in our daily lives, and how is it useful to the living organism? So we looked at diffusion in details. We went ahead and looked at the factors affecting the rate of diffusion, and then we also looked at the roles of diffusion in living organisms, both in plants and animals. So that was about our last class. Now, for our today's class, we are going to look at osmosis. Osmosis is the second physiological process that we are looking at today. And what do we expect to know in our today's class? Now, the same objectives that we had for diffusion, we expect to have them here. So by the end of this lesson, I expect you to, number 1, you should be able to define osmosis. You should be able, at the end of the topic, you should be able to describe the importance of osmosis and the roles of osmosis in living organisms. Now, osmosis tends to be a bit wider as compared to diffusion, because here we are also going to go ahead and look at some of the applications, or how osmosis is applicable in the bodies of living organisms. And so we are going to try and look at water relations in plants and in animals. Water relations in plants, water relations in animals. And we will also be looking at some of the terminologies that are usually used in osmosis. For instance, we will look at what is osmotic pressure. We look at Target pressure. We will also look at wall pressure, and we will also be able to look at other terms like crenation, plasmolysis, deplasmolysis, and we'll also be able to look at various terms that are used to describe different solutions, such as hypertonic solution, hypotonic solution, and isotonic solution. Okay, so those are some of the terminologies that you're going to meet here. They are not new to us. We will just try and understand them better. So those are our objectives of the lesson today. We will try as much as possible and cover everything here. But if we will not be done, we will proceed from where we shall stop in our next class. So that is our expectations for the lesson. Okay. Now, osmosis. Osmosis, as you all know, in some of your textbooks, osmosis is described as a special type of diffusion. That is what some textbooks say. For instance, if you look at KLB biology, they say osmosis is a special type of diffusion. Now, what is osmosis? In our last class, while talking about diffusion, we defined the word diffusion as the movement of molecules of a substance, or the movement of molecules or particles of a particular substance from a region where they are highly concentrated to a region where they are lowly concentrated. That is the definition for the word diffusion. So in diffusion, we saw that it is possible for particles of a given substance to move from a region where they are highly concentrated to a region where they're lowly concentrated. And remember, we said that diffusion only takes place in liquids and in gases. Why did we say that? We said that because it is, if you look at the particles of a liquid and the molecules or particles of a liquid and those of a gas, they're loosely packed. They're not held together like those of the solids. So it means that it is possible for diffusion to take place in liquids and in gases. In other words, it is possible for other particles to move in between the particles of a liquid and those of a gas. So it was important for us to note that because it is going to be relevant while looking at osmosis. Now, while talking about osmosis is the process by which solvent molecules or water molecules. Now, when I talk about the word solvent, water is a solvent. So water molecules or solvent molecules is a very important term here. So when defining the word osmosis, as you can see on my screen, I hope you can all see my screen. And if not, you can try and look at it clearly. You can try and zoom it so that you can be able to see my screen clearly. So I have projected the word osmosis on my screen there, and I believe you can all see it. Now, osmosis is defined as the process by which water or solvent molecules move from a region where they're highly concentrated to a region where they're lowly concentrated through a semi-permeable membrane. Now, the definition on my screen says, this is the process by which solvent or water molecules move from a lowly concentrated, or that is a dilute solution, to a highly concentrated solution across a semi-permeable membrane. Now, someone is about to ask me, or someone is just about to say that the definition that I'm putting on my screen is not right. But I want to tell you that the definition you're seeing there is very right. Let us look at it again. We're saying that this is the process by which water or solvent molecules move from a lowly concentrated or dilute solution. Now, a dilute solution is that solution which has more of the solvent molecules or more of the water molecules. So back in your mind, you need to know that when talking about a dilute solution or a lowly concentrated solution, that means that that is a solution which has a high concentration of water. So it is right for us to say that this is a process by which water or solvent molecules move from a lowly concentrated solution to a highly concentrated solution across a semi-permeable membrane. It is the same as saying the process by which solvent or water molecules move from a region where they're highly concentrated to a region where they're lowly concentrated across or through a semi-permeable membrane. Are we together? I hope we are. Now, when we say across a semi-permeable membrane, it is the same as saying through a semi-permeable what? Membrane. For instance, look at my hand, if this is the membrane, if this is the membrane, if that is the membrane, then across would mean it comes from this side to this other side. Or across, the word across and through, they mean the same thing. Across a semi-permeable membrane or through a semi-permeable membrane, that would still be the same. So let me repeat the definition once again before we proceed. We can define osmosis as the movement of water or solvent molecules from a region of their high concentration to a region of their low concentration across a semi-permeable membrane, or it can be simply defined, as you can see on my screen, as the process by which water or solvent molecules move from a lowly concentrated or dilute solution to a highly concentrated solution across a semi-permeable membrane. Now, let me just try and make you understand what we mean by the word dilute solution and concentrated solution. Now, it is important for us to understand what a dilute solution is, because as we'll be proceeding with the lesson, we will keep on meeting these two terms, dilute solution and concentrated solution. Now, a dilute solution is a solution which has less of the solute particles, and more of the solvent particles. So I am bringing other new terms, that is solvent and solute. I know you learned this in your chemistry lessons. Now, a solute is a solid which is dissolving in a liquid, and a solvent is the liquid in which a solute is dissolving. Are we together? So when we say a dilute solution is that solution which has very few particles of the solute and more of the solvent molecules of particles. And when we talk about a concentrated solution is that solution which has very less solvent molecules and more of the solute molecules. Now, I hope we are together up to that point. So we can proceed and look at my next slide here. Now, the next thing that I want us to look at now, without wasting much time, because I think I've made it clear to us what we mean by a concentrated solution and a dilute solution. Now, I want us to just highlight how to demonstrate osmosis using a visking tubing. A visking tubing is a new term to us, but I believe we all know what a visking tubing is. In one of your practicals in Form 1, you did an experiment demonstrating osmosis using a visking tubing. Okay, is it possible to hear me? Someone said that I'm not audible, so is it a problem to everyone? Can you hear me? Now, James, could you please nod your head if you can hear me? Thank you very much. I am now sure that everyone can hear me. Now, I was saying, visking tubing is familiar to all of us, and at one given point, you all saw what a visking tubing is in one of the experiments that you did in Form 1. But now we are going to, I want us just to see, to have some highlights of what a visking tubing is and how it is used to demonstrate osmosis. Now, osmosis takes place in both the living tissues and in the non-living tissues. Now, the living tissues include the membranes, the membranes that we have within the bodies of organisms, both in plants and animals. You remember in our last class, I told you what a membrane is. And we said that a membrane is a surface structure which encloses the contents of a cell. A surface structure that encloses the surface of a cell. And we listed some of the examples of membranes, and we said that membranes are named according to the structures which they enclose. For instance, when you talk about a cell membrane is a surface structure which encloses the content of a cell. We looked at what a tonoplast is, and we said that a tonoplast is a surface structure which encloses the content of the vacuole. We looked at the ribosomal membrane. We looked at the mitochondrial membrane. We looked at the membranes of a chloroplast. So visking tubing is a semi-permeable membrane that is artificial. That is all about it. An artificial semi-permeable membrane that we use in our laboratories is a visking tubing. So we say that it is a semi-permeable membrane because it shares almost those physical. Okay. I'm sorry. Now we are saying that a visking tubing is a semi-permeable membrane because it shares some of the properties of a cell membrane. Now, visking tubing, just like the cell membrane, is semi-permeable. It has some tiny pores that can only allow particles or substances of small size to pass through it, just like a cell membrane can do. So, a visking tubing is selectively or partially permeable, just like the cell membrane. So, osmosis is the movement of water or solvent molecules from a lowly or dilute solution to a highly concentrated solution across a semi-permeable membrane. Now, in this experiment that we're just going to see here, but we won't conduct it physically, we are going to see how we can demonstrate osmosis using a visking tubing. So what you're going to do, assuming that we have a beaker which is filled with about 350 cubic centimeters of water. So I am already at the procedures you can see on my screen. And then we are provided with a visking tubing. So when using a visking tubing, now visking tubing is usually opened on both sides. It is a tube. It is a tube-like structure, but it is semi-permeable. And it is always opened on both sides. So when using a visking tubing, what you do, one end you will tie using a rope or a string or whichever substance that is provided for you to tie the visking tubing. So what we'll do is that for you to do that, you first have to moisten it, and we are saying that dip one end of the visking tubing provided to moisten it, and then open the visking tubing and tie one end with the thread provided. So in the next slide you're going to see here, we are already going to see okay, before we look at the visking tubing, now once you have tied the visking tubing, one end of the visking tubing with a thread, you fill the visking tubing halfway. Fill the visking tubing with salt. Just a minute. Now, fill the visking tubing with the salt solution provided, and then tie the open end of the visking tubing. Now, ensure that there is no salt solution that spills from the visking tubing. Then immerse the visking tubing into the distilled water in the beaker and suspend it as shown in the diagram below. And this is the diagram. Look at this now. This is our diagram. So you did something which is similar, which is similar to this in one of the experiments you did in Form 1. And this is a beaker which contains water. And this is our visking tubing. And as you can see at this point, I hope you can all see look at my cursor on the screen. This is the one of the ends of the visking tubing is tied using a rope. And then the visking tubing here is filled halfway with a salt solution. So the solution inside the visking tubing is a salt solution. Now, a salt solution simply means salt that has been dissolved in water. So salt that is dissolved in water is a salt solution. So the salt solution is filled inside the visking tubing halfway, and then this other end, the other end of the visking tubing is also tied so that it does not spill out of the visking tubing. So this is what we expect. I mean, this is what we have: water inside the distilled water. Distilled water is water which is pure water without any kind of solute in it. So pure water or distilled water is filled inside the beaker, and now you can see what we have here. So visking tubing is already here with us. So now we would expect you to record your observations after 30 minutes. Now, maybe I can ask one of us to tell us what do you think you'd expect after 30 minutes, and you should be able to explain your observation. So what do you expect after 30 minutes of the experiment? Let me find out who will explain this to us. Somebody by the name Moses Carissa. Moses Carissa, are you there? Now I will unmute you for a minute. Moses Carissa, are you there?
Yes.
Great. So could you please tell us what you expect to observe? What is your expectations from this experiment after 30 minutes? What would you expect?
So we'll observe that water entered inside the solution through the semi-permeable membrane. So we can say that more water entered in the visking tubing.
That is after 30 minutes, right?
Yes.
Great. So Moses Carissa expects to see. Okay, Moses, I will mute you. I will mute you just a minute. Okay, great. So we expect that after 30 minutes, more water will move from the beaker and get into the visking tubing across or through the semi-permeable membrane of the visking tubing. Great. So who will explain to us this observation? Why do we think that more of the water molecules will move from the beaker into the visking tubing through the semi-permeable membrane? Great. So I'm seeing very many hands have been raised. So I will, great. Gideon Simon.
Yeah.
Gideon Simon, are you there?
Yeah.
Great.
I'm here.
Yes. So could you please explain to us the observations?
So more water will move from the beaker to the
more water will move from the beaker into.
Okay. So more water will move from the beaker to the visking tubing because
Okay, you are not really very clear, but I believe I've gotten your point. So Let me just mute you. Or you can mute yourself, kindly. Now, without wasting time, I believe we all have the idea because I can see a good number of people have raised their hands, meaning we all know what we expect. But now, just to explain it in simple terms, just to explain this in simple terms. Now, we have said that we expect to see, there are two main observations that we expect here. And for your information, in a practical exams that I think we are going to be meeting in front here, we will be asking you state any two observations, because we expect you to tell us at least two observations from this. Now, number one, what you expect to see, we expect to see the level of the salt solution in the visking tubing to increase, or the visking tubing will tend to swell or increase in size. More of the water will be getting into the visking tubing from the beaker. And that would mean that the level of water in the beaker will reduce. So those are two observations. One, the level of the salt solution will increase, and therefore the visking tubing will swell, or it will enlarge. Number two, the level of water in the beaker will decrease. So this clearly tells you that when we talk about a distilled water, that is pure water. So osmosis will take place so that water will be moving from the distilled water, where its concentration is high, to the salt solution inside the visking tubing, where its concentration is low, across or through the semi-permeable membrane of the visking tubing. So you observe, that should be your explanation for the observation. You should simply say that water moved through osmosis from the distilled water in the beaker, where its concentration is high, to the salt solution inside the visking tubing, where its concentration is low, across the semi-permeable membrane. That is the explanation. So that tells you that osmosis took place, and therefore water moved from the region of its high concentration to the region of its low concentration across the semi-permeable membrane into the visking tubing. Great. Now, Ernest Mumo has a question. I'm just trying to. Ernest Mumo, could you please type the question on the chat so that I can be able to find it? Just write it there on the chat. I'm trying to find you, but because the number is very large, I am unable to just see you here. So just type the question, I'll be able to see it. Ernest Mumo, thank you. Now we can proceed as Ernest is working on the question. So we expect that after 30 minutes, water will move from the beaker where its concentration is high to the salt solution where its concentration is low. So now you can see that the highly concentrated solution here is the salt solution, while the lowly concentrated solution is the distilled water, which sincerely has got no solute in it. But when you're talking about the concentration of water in terms of water concentration, the distilled water is highly concentrated with with while the salt solution is lowly concentrated with water. So osmosis, as we said earlier, occurs from the region with a high concentration of water to the region of low concentration of water. And that is what you have seen there from that setup. Great. Now, Ernest is asking, when do we use the terms hypertonic and hypotonic? So like I said, we are going to be able to understand these terms later on. I will introduce them. But still, just to highlight what they are, a hypertonic solution is a solution which has got a high concentration of the solutes. It is in what is it? Hypertonic solution, hypertonic solution is what is always referred to as a highly concentrated solution, which means it has got a high concentration of the solutes than the solvent. Now, hypotonic is the opposite of hypertonic. And that means that hypotonic solution is that solution which has got a low concentration of the solute particles, but high concentration of the solvent what? Particles. We are going to look at them later on, so do not worry. But we can also, well, I think Ernest Mumo is asking the question because of the setup here. So in this setup, we can assume, or we can say that the salt solution is the hypertonic what? Solution, while the distilled water is the hypotonic solution. So the salt solution is our hypertonic solution, while the distilled water is our hypotonic solution. Thank you. So those who are raising their hands, I can see Ryan Kim Jr. has raised his hand. Could you please just use the charts? We can proceed meanwhile. So we can, if you're asked to record your observation, just write the observations that I've mentioned, and you can also explain. So, and from the diagram, we can say that now the visking tubing is semi-permeable, and therefore it only allows the small water molecules to pass through, but not the large salt molecules. So that clearly tells you that salt molecules are large, and therefore they cannot escape from the visking tubing into the beaker. But instead, it is the water molecules which will pass through the visking tubing from the beaker. So the water molecules move from the distilled water in the beaker, where their concentration is high, across the visking tubing into the salt solution. And the salt solution for this case is highly concentrated, and it is said to be hypertonic. So the water molecules keep moving until the visking tubing becomes turgid. Now, the word turgid is now a new or a strange term that we are introducing there. Now, can you say osmosis is the movement of water from the region of low solute concentration to the region of high, to the region of high solute concentration across concentration gradient? Now, here we don't talk about the word concentration gradient, but I think you meant across a semi-permeable membrane. So you can say it is the movement of water molecules from a region of low solute concentration, which is just like from a lowly concentrated what? Region. That is what we say. So that is right. But you don't use the word concentration gradient there. You use across a semi-permeable membrane. So Kim Jr. is asking, so you can say that the salt solution is hypertonic to the distilled water. Hence, distilled water moved into the visking tubing through osmosis. Very good. That is very right. You can say that. So the salt solution is a hypertonic to the distilled water, meaning it is highly concentrated. It is more concentrated than the distilled water. And I started by saying that the distilled water can also be said it is pure, because distilled water is that water which has got no solute in it. Haina kitu ndani yake. Ni maji, pure water. Okay? Great. So we're saying there that my last point there, the water molecules keep moving until the visking tubing becomes turgid. Great. Let us proceed. Now, the lowly concentrated solution is said to be hypotonic, and I think I just mentioned this a few minutes ago. The lowly concentrated solution is said to be hypotonic. Now, a hypotonic solution is that solution which is lowly concentrated. It is dilute. This is a solution which has got very few solute particles in it. For example, if you have a glass of water and then you put a few particles of sugar in it, and then you taste, you will not feel the taste of sugar. Why? This is because the amount of sugar particles in that particular solution is very low. So that is why we're saying that this water is dilute, meaning it has got very few particles of the solute in it. Now, the opposite of the word hypotonic, like I said, let me skip the second point there, we can look at it later. The highly concentrated solution is said to be hypertonic. So if you're comparing two solutions, one which is lowly concentrated and one which is highly concentrated. Now, the one which is highly concentrated is said to be hypertonic, sorry, is said to be hypertonic solution, while the one which is lowly concentrated is said to be hypotonic solution. Okay, great. Now, someone is asking, but the lowly concentrated solution can have no solute at all in our example. Yes, a lowly concentrated solution, we only says it is lowly because when we're comparing two solutions, it is found to be having very few solute particles in it. It can be having very few or none. Okay, so that is a lowly concentrated solution. Like we have looked at we've given an example using the pure or the distilled water in that experiment above there. Okay, great. Now, in relation to just a minute. Okay. So I'm working on my internet. I just lost my internet, but I hope we are there. Just trying to recharge it, check if it's a bit unstable here. Okay. Okay. Now we are told that in relation to a plant cell, hypotonic solution is less concentrated than the cells have. Now, don't worry about this. This is going to be explained better in front when we'll be looking at the water relations. Okay. Now, let me just take you back to this diagram here. Before we just say that there'll be net movement of water molecules from the beaker to the salt solution. But now, when you go ahead here and look at, now we are introducing a new term, which is isotonic solution. Now, an isotonic solution, this is a solution, or these are solutions that have the same concentration. These are solutions that have the same concentration. So let's say if you have got, I was giving an example using a glass of water, which one has got few sugar particles in it, and one has got more sugar particles in it. So this one with more sugar particles in it will be said to be a hypertonic solution, and the one with low sugar particles in it is said to be a hypotonic solution. So if you have two solutions which have got an equal concentration, then we say that they are isotonic what? Solution. So isotonic solution is simply a solution which has got an equal concentration. Okay? Now, what if they ask for a control experiment? We are going to, there's an experiment, I think another experiment you're going to look at, which I will be able to explain what an control experiment is. And then is osmosis. In osmosis, we use water molecules. What about in diffusion? Now, thank you. I think those are some, it is important to ask such because I know they bring a lot of confusion, especially when you are in an examination situation and you are told to define osmosis and diffusion. Students always wonder, should I say water or should I say solutes? Now, osmosis, to be specific, osmosis is only dealing with solvent molecules or water molecules only. We don't, when solids are moving from one region where they're highly concentrated to another one where they're lowly concentrated, that is not osmosis, my friend. Osmosis is purely about water or solvent molecules moving from a region where they're highly concentrated to a region where they're lowly what? Concentrated across a semi-permeable what? Membrane. When you're dealing with diffusion is about particles of a particular substance. Particles or molecules of a particular substance. They can either be solids or they can either be liquids, moving from one region where they're highly concentrated to another region where they're lowly concentrated. Okay? Yeah. Great. So I think I've answered that. The person was asking about using water in osmosis and not in diffusion. I think you've understood that. In osmosis, we're only talking about solvent molecules or water molecules. The reason we are saying solvent, a solvent can be any liquid that has the ability to dissolve a particular solute. It must not necessarily be water, but in some books you'll find them strictly talking about water particles. Okay, so osmosis is purely about solvent or water molecules. Now, can you use other solvent other than water in osmosis? I think I've just explained that, Vincent Kinywa. Thank you. So, as we proceed, now what you're going to look at next are some terminologies that are usually used in relation to osmosis. And this is osmotic pressure and osmotic potential. Now, it is important for us to understand osmotic pressure and osmotic potential because they are very key terms that are used while looking at the water relations in plants and in animals. Okay? Now, the word osmotic pressure, I think we have all heard about osmotic pressure in very many cases. And I want us to look at what is this osmotic pressure about and what is osmotic potential. Now we'll start by looking at osmotic pressure. Now, when we have, in the experiment that we just looked at a few minutes ago, we saw that it was the water molecules moving from a region where they are lowly concentrated or from the dilute solution to the salt solution, which is highly concentrated, and that is inside the visking tubing. So, osmotic pressure here, as you can see, it is defined as the pressure or force that a concentrated solution develops to draw water molecules from distilled water through a semi-permeable membrane. If you look at your books, I think it is scale B. Yeah, it is scale B that says that osmotic pressure is the pressure that is developed by a concentrated solution so that it can draw water from a dilute solution across a semi-permeable membrane. So the same thing is just explained here, but other books will use different terms, but the same thing, it is the same thing. So we are saying that osmotic pressure is the pressure or a force that a concentrated solution develops to draw water molecules from distilled water through a semi-permeable membrane. So assume you have two solutions which are separated by a semi-permeable membrane. Two solutions which are separated by a semi-permeable membrane. So this osmotic pressure, I'm just trying to find the experiments that I can use the diagrams there to explain this. Great, look at this. We have two solutions here. One, we have the dilute solution, which is the distilled water. And then we have the concentrated solution, which is the salt solution. So we have concentrated solution inside the visking tubing and a dilute solution inside the beaker. Now we are going to use this diagram here to explain the osmotic pressure. So we have said that the osmotic pressure is the pressure or a force that a concentrated solution develops to draw water molecules from distilled water through or across a semi-permeable membrane. So that pressure that this in this diagram here, the salt solution is the concentrated diagram. So the pressure or the force which is developed by the salt solution so that it can draw water from the distilled water inside the beaker is the osmotic pressure. So the pressure that is developed by a concentrated solution so that it can draw water from a dilute solution across a semi-permeable membrane is what is called osmotic pressure. I think I have some questions here. Now, what is the difference between osmotic pressure and osmotic potential? Now, I said I'm going to explain that. Don't worry about osmotic potential, please. Should you say a highly concentrated solution draws water or the concentrated solution draws water in osmotic pressure? Now, what is the difference between highly concentrated solution and a concentrated solution? They're the same thing. A highly concentrated solution is the same as a concentrated solution. The opposite of that is a lowly concentrated solution or a dilute solution. Now, highly concentrated solution, opposite to that is lowly concentrated what? Solution. A concentrated solution, the opposite to that is dilute solution. It is the same thing, Clinton, okay? Yes. So we are at osmotic pressure. We have said that this is the pressure or a force that a concentrated solution develops to draw water molecules from distilled water through a semi-permeable membrane. Now, in the second point that it is said, it is the pressure that can be applied against a semi-permeable membrane to stop osmosis from taking what? Place. It is the pressure that can be developed against the semi-permeable membrane to stop osmosis from taking place. Now, assume more of the concentrated solution has absorbed, has drawn water from the dilute solution. It reaches a point whereby now this solution, some pressure is now developed against the semi-permeable membrane so that more of the water molecules are not drawn. So it reaches a point whereby once more of the water molecules have gotten into the concentrated solution, osmosis will have to be stopped by applying some pressure or some force against the semi-permeable membrane to stop the water molecules from getting into the what? Into the concentrated solution. Now, at that point, the concentrated solution shall have been dilute because more of the water molecules shall have gotten into it. So we'll be assuming that it has now become dilute. It's just like adding more water into a concentrated tea so that it doesn't become concentrated, but now it becomes dilute. So that pressure that is applied against the semi-permeable membrane so that osmosis can stop is also the osmotic pressure. Now, our teachers told us that osmotic pressure is the force by semi-permeable membrane to stop water molecules from hypotonic solution entering the hypertonic solution. Yes, that is true, David Gachema. It is the same thing that I've just explained. Okay? It is the same point. It is outlined in my second point here, that the pressure that can be applied against the semi-permeable membrane to stop osmosis from taking place. Now, it is that pressure that will be applied against the semi-permeable membrane to stop more water molecules moving from the dilute or the hypotonic solution so that they cannot get into the hypertonic what? Solution or the concentrated what? Solution. So if you have two solutions which are separated by a semi-permeable, once again, a semi-permeable membrane, if you have two solutions which are separated by a semi-permeable membrane, now, there is that pressure that can be applied against the semi-permeable membrane so that water molecules are stopped from moving from the hypotonic solution into the hypertonic what? Solution. And that pressure is called the osmotic pressure. Let me find out these questions here. And could you please? Okay. Teacher, how can it stop osmosis, yet it is used to help distilled water to move to the salt solution? Now, I said when osmosis is taking place, in other words, when water molecules are moving, from the highly concentrated, from the lowly concentrated solution or from the dilute solution into the highly concentrated solution, this concentrated solution is the one that develops a pressure. Now, this pressure will perform two functions. Number one, this pressure here is developed so that it can water molecules can be drawn from the distilled solution across the semi-permeable membrane into the concentrated solution. So there'll be that net movement of water molecules from the dilute solution into the concentrated solution until these two solutions have become isotonic. In other words, until these two solutions have acquired an equal concentration. So therefore, it will mean that once the two solutions have become isotonic, or once they have had an equal concentration, some force will be applied against the cell membrane or against the semi-permeable membrane to stop water molecules from moving from one solution to the other, or to stop osmosis from taking place. So to answer these questions that I'm now seeing here, I think that is what was not coming out clearly. Let me start by looking at the question that was asked by someone who is not using the official name here, but is asking, what is the difference between osmotic? Okay, Clinton. Clinton said, should you say highly concentrated? No. David. Is it David O'Brien? Just a minute. I'm trying to find it on my charts here. Membrane to stop the molecules entering the hypotension. The teacher. Just a minute, kindly. Is osmosis? Can you use osmosis? Can you say osmosis is the movement of water molecules? That is from Clinton. Now, is the pressure, okay, Richard, I think it is Richard. Let me start from Lemi Mundia, who was asking, who asked me to repeat it. I think I've repeated it. Lemi, could you please, Lemi Mundia, I think, could you please confirm if you've understood it clearly? Then Richard Onjo is asking, I think the pressures, I think it will be applied when two solutions become isotonic. Is it? Yes, that is what I've just explained, Richard. I said that once we have had an equal concentration of water molecules, or once the two solutions have become isotonic, I said that isotonic solution is a solution which has got equal concentration. Okay, so once the two solutions have become isotonic, a force will be applied on the semi-permeable membrane to stop osmosis from taking place, and this force is what is called the osmotic pressure. So like I said, when we have a concentrated solution, this concentrated solution, it develops a force or a pressure, and that force or pressure is also referred to as osmotic pressure, which draws water from the dilute solution. Okay, the osmotic pressure again here, we've said that it is developed. It is that force or pressure that is developed to stop osmosis from taking place. And when will osmosis stop? Osmosis will stop once the two solutions have become what? Have become isotonic. Okay. Then that was a question from I think that was a question from Richard Wanjoy. So Richard, could you please confirm if you have understood or you still need an explanation? Then Ernest Mumo is asking again, so does it mean that there are two types of osmotic pressure, or osmotic pressure has two functions? Now you can say it performs two functions. One, it is developed so that a concentrated solution can draw water from a dilute solution. Then two, it is developed to stop osmosis from taking place once we have acquired an isotonic state. Okay, that was Ernest Mumo. If you have understood, you can let me know. And then Willie is asking, it is also the ability of a concentrated solution to draw water by osmosis from another solution separated by a I think that is a repetition, and I've explained that. Ernest Mumo is asking, so does osmotic pressure? I think I've explained that, Ernest Mumo, and I have responded to all the questions. Okay, thank you. Thank you. I think the rest are saying that they've understood, and I think we can continue without wasting much time. Thank you for that. And sorry for looking too much into the questions. We can now proceed. The next one that we're looking at now, the next terminology here in my next slide, now to demonstrate osmosis using potato cylinders. This one, I think they're not going to look at it. But our potato cylinders are, I don't know whether you did this or you did the other experiment. Now there's a way in which you can make a potato cylinder by just peeling a potato to make it look like a cylinder. Now the potato here is used, the potato here has got living tissues which will act as our cell membrane, so which is semi-permeable, so it will allow for that movement. I will not look at this because it is actually, this is very, very practical, so I may not really look at it. Okay. Great. Osmotic potential. Osmotic potential. Now, before I look at osmotic potential, someone is asking another question here. Now, Adam Gatari, I think we are now moving to the osmotic potential. We had not really looked at it. Okay. Now, osmotic potential is our next terminology here. Now, osmotic potential. Now, in different books, will give you different meanings. Will give you the same meaning, but using different terms. For instance, the one that is commonly used by you students is KLB. And I think KLB puts osmotic potential as the hidden, the hidden pressure or the hidden force that only becomes real when a concentrated solution is separated from a dilute solution using a semi-permeable what? Membrane. Okay? But now, what the concept is, it's a very simple thing here. On my screen, you can see we're saying that it is a measure of the tendency of a solution to withdraw water molecules from pure water through a semi-permeable membrane. Now, someone is just about to say, but Peter, is this the same thing as osmotic pressure? No. Let us look at it again. We're saying that osmotic potential is the measure of the tendency of a solution. You get the measure of the tendency. It's like the ability. Now, from your English knowledge, you know, potential refers to that hidden ability. People who have talent, you've always been told that, you know, you have got a lot of potential, meaning it is some hidden ability in you that we can see. Or if you come to class and then we see you, people who have got talents in doing other things in class, we always tell you that, you know, we can see some potential in you. So the potential is like the hidden ability, some hidden ability in you, you get? So when we talk about osmotic potential, it is like that hidden force or hidden pressure. It will only become real when that concentrated solution is separated from a dilute solution using a semi-permeable what? Membrane. So it is the osmotic potential is what leads to osmotic pressure. Now you get the concept. It is the osmotic potential that leads to osmotic pressure. That is why we're saying that it is the measure of the tendency of a solution to withdraw water from water molecules from pure water through a semi-permeable membrane. It is the ability of a solution to take up water by what? Osmosis. Very simple definition in my second statement here. It is the ability of a solution to take up water by osmosis. It is not a force, or it is a force that only becomes real. Students, if I just had a better way to explain this, I would use the simplest words. But I think the simplest words to use here is it is the ability of a solution to take up water by osmosis. Okay? It is like an ability, hidden potential. Let me look at I think a question here. Excuse me, teacher. While defining osmotic potential, can we say it is hidden energy that only comes true when a concentrated substance is separated from distilled water by a semi-permeable membrane? Yeah, this is Galaxy. Galaxy Co-Prime is what your name, okay? Try and use your official name so that we can know who is asking the question. So I think I will ignore questions which are coming from students who are not using their official names. Please explain more. I haven't understood. This is Ernest Momo. Okay, let me just repeat the same point so that we can continue and no one is left behind. We are saying, we're talking about osmotic potential. And I started by using an analogy of what we always see in our daily lives. Now, if you're told, you know, you have got a lot of potential in you. When somebody tells you have a potential in you, that means that you have got some hidden ability, like a talent that we are seeing that can only become real when you start applying it in the daily life. So that is why we think that when we talk about the word osmotic potential, we think that osmotic potential is the hidden force or pressure that only becomes real when a concentrated solution is separated from a dilute solution using a semi-permeable what? Membrane. It becomes real. And why did it become real? Because these are two solutions which have got different concentrations. One is highly concentrated and the other one is lowly concentrated. So Ernest Momo, in simple words, we can say that osmotic potential is the ability of a solution to take up water by osmosis. And if we have two solutions where one is highly concentrated and the other one is lowly concentrated, then we can say that the solution which is highly concentrated, it has got a high osmotic what? Potential. Why do we say it has got a high osmotic potential? We are saying that it has got a high osmotic potential because it has got the ability to draw water from a dilute solution. So you need to know that osmotic potential is what leads to osmotic pressure. Okay? I hope I have made myself very clear. Now, can we say that osmotic potential is ability? Can we say that osmotic potential is the ability of water molecules to move from a region where they are highly concentrated to a region where they're lowly concentrated across a semi-permeable membrane? In biology today, they say that osmotic potential is the ability of water molecules to move from their high concentration to the. Okay, I think the definitions that that some of you have asked here, they're just right, except that now it is the choice of words that people use. Okay. But now I think the best thing that you need to know is that, and I need you to get this from me as a teacher. And there have always been very many words that have led to confusion. But again, as a student, we always insist that use the simplest words to describe a particular definition. Do not get confused and look for some terminologies that may end up confusing you yourself. In simple terms, if you've been listening to what I've been explaining here, and I want to repeat this once and for all, and I hope you're going to be able to understand. We are saying that when talking about osmotic potential, potential is an ability, okay? And do not confuse osmotic potential and osmotic pressure. We say that osmotic pressure is that pressure or force which is developed by a concentrated solution to draw water molecules from a lowly concentrated or from a dilute solution across a semi-permeable what? Membrane. And it is also that pressure that is developed, that force that is developed to stop osmosis from taking place. That is for osmotic pressure. Please understand it that way and do not confuse it with osmotic potential. So when talking about osmotic potential here is now being the ability. It is being the ability of water molecules, the ability of a solution to take up a solution, sorry. It is the ability of a solution to take up water by osmosis. I'm very sorry for that. Now, it is like the measure of the tendency of a solution to withdraw water molecules from pure water through a semi-permeable membrane. So let me just explain this using the simplest way. If you have a highly concentrated solution, and a lowly concentrated solution, you find that the highly concentrated solution is the one which has the ability to draw water molecules from that dilute what? Solution. Let me just take you back here. Let me just take you back to this diagram that we're using here for experiment, okay? We have two solutions here, students. The first solution here is the salt solution, which is inside the visking tubing. And the other one is the dilute solution, which is inside the beaker, okay? So these two solutions are sorry, I was looking at some of the questions there again, but I think they're just confusion of terms, and I will make it clear, I think, using this experiment here. So we have two solutions here. One, the salt solution there, and the other one is the distilled water in the beaker. So you need to know that the salt solution here is the highly concentrated solution, while the distilled water is the lowly concentrated what? Solution. So when talking about osmotic potential, students, you need to note that osmotic potential is only, is the ability of, is the ability of a concentrated solution to draw water from a dilute solution. So we are saying that the salt solution there, because it is the concentrated solution, it has got a high osmotic what? Potential, and therefore it has the ability to draw water from the distilled water, which is a lowly concentrated what? Solution. You understand? Now, the distilled water or the dilute solution has got a low osmotic potential. In fact, it may not even be having osmotic potential, because if it is lowly concentrated, it is unable to draw water from a highly concentrated what? Solution. So osmotic potential is only applicable when talking about that ability of a concentrated solution to draw water from a dilute solution. So maybe I think I have tried and used the simplest words possible. And like I always say, students, if it is still a challenge to you, we are always available. I will always be there for you. Whenever you need me, you can try and contact me. A good number of the students, I think, have our contacts. Just try and ask me, or you can reach out to any biology teacher and they'll be able to share with you. Yeah, Samson Moria, it is right. It is right to say that when a solution has got high osmotic pressure, then it means that that one with the high osmotic pressure has got a high osmotic potential, okay? Yeah, thank you. I think they're just words that are now ringing into your mind, and now you feel like, is this right? Do not confuse yourself. Relax, okay? I always tell you people, relax. Take everything simple. Do not panic. Take it as it is, okay? Now, this is just what you're seeing on my screen here is just but an experiment here, an illustration of some experiment here. This is okay. Maybe we should look at this. Okay, I didn't mention about osmometer. Now, osmometer is an instrument that is used to measure the osmotic pressure of a solution. It is called osmometer. It is an instrument which is used to measure the osmotic pressure of a solution. Okay, Ernest Mumo. Oh, okay, please try and contact any member from 2 Southwest, and they'll give you my number. Okay. Great. So, if there are any other questions about the same, you can read more into the books. Try and look at, now that even at home and you can easily access the internet. Please try and look at it. The more you look at them, try and see what other sources say. I think there may be other simpler terms that we can easily understand. I think there's this question that I didn't see from Victor, that can you say that osmotic pressure is the force that acts against osmosis at equilibrium? Yes, that is right. That is right. I mean, it's the same thing that I've been saying. It's the same thing that I've been saying. Great. So let us proceed. There is this experiment here, but it is not really coming out clearly. Or maybe let me just try and show you what it means. Now we have a solution here, and we have a pure solvent here. A solution is made up of a solute and solvent. Okay. Then pure solvent solution, pure solvent solution. This experiment, like I said, now look, this is what I want you to this is what I want us to analyze. Okay. So if you look at my diagram here, this is a diagram that is made up of two solutions. On where I'm showing my cursor here, this is where we have got a concentrated solution or a hypertonic solution. This is where this other side, we have got a dilute solution or hypotonic solution. Now, for those of you who didn't understand what hypertonic and hypotonic solutions are, you can now see. When you're talking about hypertonic solution, that is a solution which is highly concentrated. While when talking about hypotonic solution, that is a solution which is lowly concentrated, or the dilute solution. So just comparing the two, you should note that a dilute solution has more water molecules. As you can see, the water molecules, the blue dots here represent the water molecules. Dilute solution has got more water molecules, and it has less solute molecules. The solute molecules are the yellow ones here on this side. And then it has got a low osmotic pressure. And it is separated, these solutions here are separated by a semi-permeable membrane. So this side where we have the hypertonic solution or the concentrated solution, here we have got less water. In fact, you can even count them there around 7 of them. And then concentrated solution has got more solute particles. You can see the solute particles are very many here. And then we're saying that it has got a high osmotic pressure. Okay, great. Now the next thing that I want us to look at is that is now the water relations in cells of plants and animals. Water relations in plants and animal cells. A very interesting area. Very interesting area. Very common in examinations, not only in school, but even at the national level. If you've been looking at the past KCSE papers, this is one area that you'll never miss. I'm not saying that other areas will miss, but this is always a very sure bet. It is very interesting to understand this. Okay. Oh, okay. I'm trying to work. Alvin, could you please? Is it possible to hear me now? Now, water relations in cells. When we talk about water relations, this refers to the movement of water in and out of the cell and the effects of such movement on the cell. In other words, when we have a plant cell and an animal cell, so water relations here means the movement of water into the plant cell and into the animal cell, and out of the plant cell and out of the animal cell, and the effect. What effect does it have? What effect does this movement of water into and out of the cell have? So when you talk about water relations, it simply refers to the movement of water into and out of the cell and the effects of such movement on the cell. So we'll start by looking at water relations in animal cells. Water relations in animal cells. I decided to use these simple diagrams here so that we can know what it means when water moves into and out of a cell. So we will start by looking at water relations in animal cell. And for the animal cells, we are going to use just an example of an animal cell. And for our case, we are using red blood cells in animals. Now, red blood cell is an example of an animal cell. Okay? Red blood cells is an example of an animal cell. So we know that the cell, an animal cell, as we know what it is made of. There are those cell structures or organelles which are only found in the animal cell and not in the plant cell. For example, animal cells have got cell membrane. The plant cell have got both cell membrane and the cell wall. You know that, okay? The cell membrane of the animal cell is almost the same as the one for the plant cell in terms of their properties, in terms of the properties that they share. So the plant cell, the animal cell has got a vacuole in it. This, which is made up of solutions, salt and sugar solutions which are found there. There are very many solutes which are found or contained in the vacuoles of the animal cell. Now, if we now I want to try and make it simple by comparing what would happen if an animal cell is placed in a hypotonic or a distilled water, hypotonic solution or a distilled water. This diagram here. So this is a plant cell. This is an animal cell placed in a hypotonic solution or distilled water. Then here, this is an animal cell placed in a hypertonic solution, which is a concentrated what? Solution. Okay. Now we'll start by looking at what will happen to an animal cell when it is placed in a hypotonic solution or in a distilled water. What will happen to an animal cell when it is placed in a hypotonic solution or in distilled water? So look at this. When an animal cell is placed in a hypotonic solution or distilled water, now water will be drawn into the cell by osmosis. In other words, because the animal cell contains certain solutes, we say that it is highly concentrated. So the vacuole inside the animal cell is highly, the cytoplasm, inside the cytoplasm of the animal cell contains, we have got very many solutes in it. And so you will realize that inside there, because of the solutes which are contained inside the cytoplasm, it makes the animal cell to be to be highly concentrated. So when it is placed in a hypotonic solution or a solution which is lowly concentrated, this animal cell develops an osmotic pressure. So it draws water from the hypotonic solution through osmosis across the semi-permeable cell membrane. So what happens when water gets into the animal cell by osmosis? The cell will swell. It will swell until it bursts finally. So in the last diagram here, this shows a cell that is bursting. This is a cell that is bursting. So the cell swells until its membrane bursts. So the bursting of the red blood cells or the bursting of an animal cell when it is placed in a hypotonic solution or in a distilled water is called, that process is called hemolysis. Hemolysis is the name of the process. In other words, hemolysis is the process, is the bursting of red blood cells when it is placed in a hypotonic solution. That is hemolysis, the bursting of red blood cells. And as you can see, this diagram here illustrates that. Now, let us see what happens when a red blood cell or an animal cell is placed in a hypotonic solution. Now, a hypotonic solution is a solution which is highly concentrated. So for this case, when it is placed in a hypertonic solution, that would mean that a solution which is highly concentrated than that animal cell. We are not saying that the animal cell is not does not contain solutes, and so it is not concentrated. No, the animal cell is also just concentrated. I mean, the cytoplasm and the vacuole inside the animal cell is also concentrated using because of the presence of certain solutes. But then, when compared to this other hypertonic solution, this solution is now highly concentrated than the animal cell. So when an animal cell is placed in such kind of a solution, what happens is that water will be drawn out of the animal cell by osmosis. Now, instead of gaining water, this animal cell will start losing water. Water will be drawn out of it by osmosis into that particular hypertonic solution. So as water is drawn out of that cell by osmosis, the cell shrinks. Now, that to shrink simply means to reduce in size. Like, instead of swelling, it now reduces in size because it is losing the water. So as the cell shrinks or wrinkles, we say that it becomes crenated, and the process is called crenation. So crenation is the opposite of hemolysis. Are we together? Crenation is the opposite of hemolysis. Does hemolysis happen in the human body, and what is the effect of bursting? Okay, I will respond to that, Joseph. So now the cell shrinks or wrinkles, and then we say it becomes crenated. Now, when you're asked to define what is crenation, crenation is just simply the opposite of hemolysis, and it can be defined as the process by which the animal or a red blood cell shrinks when it is placed in a hypertonic solution, because it will now start losing the water, or the hypertonic solution, because it will be having a very high osmotic pressure. It will draw water from the animal cell, and so the cell will be shrinking. Okay? Now, because of what we have seen here, because of the crenation and hemolysis, it is always important for the body cells, the body fluids and the plasma. Are we there? It is always important for the body cells and the plasma to be kept at an equal concentration, because remember, it is inside the body fluids and the plasma where the body cells are usually suspended. Our body cells are usually suspended inside the body fluids and the plasma. For example, we know we have got the red blood cells which are suspended inside the plasma, plus other body fluids which contain other cells. So if the cells and these body fluids are not kept at an equal concentration, it means that they will either experience hemolysis or crenation. So it is always important for our body cells to to be kept at an equal concentration, so that there is no hemolysis and there is no crenation. Okay. Now, Joseph, your question, I think I have said that. Now, in the human bodies, there is a mechanism that is always put in place to ensure that the body fluids, the plasma, and even the cells are kept at an equal concentration, so that there is no hemolysis and crenation of those cells in our bodies. And more of this, we will learn them when we will get to the topic, one of the topics in Form 2 called homeostasis, and it is called homeostasis and thermoregulation. Homeostasis and thermoregulation, I think so. So that is where we learn more of the body fluids and how they interact with the cells. It is a very interesting topic that we will look at in Form 2. In the Form 2, it's one of the Form 2 syllabus. It is one of the Form 2 topics. So if I don't know whether there are any questions on this area, but maybe another diagram that is shown here, as you can see, animal cell. The animal cell, when placed in a hypotonic solution, it will burst, it will rupture. In an isotonic solution, there is nothing that happens. It is just normal in an and then when placed in a hypertonic solution, it shrinks. So this is what happens. Now we are supposed to look at what happens when a plant cell is placed in hypertonic solution, isotonic solution, in the hypotonic solution, or what is called the water relations in plant cells. But because of time, we just have a minute left. We will stop at that point. But we can start from water relations in our next class. Now, in our next class, we'll be looking at water relations in plant cells, the factors affecting the rate of osmosis, the roles of osmosis in plants and the animals, and finally, we will look at the last bit of the topic, which is the active transport. Active transport. So, maybe I need to know if there are any questions up to that point before I end the class. I don't know if there are any questions before I end the class. Now, okay, now as we come to the end of the class, I want to give out some work today. And maybe you can write them down, the following questions. Now, number one, define the following terms. Number one, Target pressure and wall pressure. Target pressure and wall pressure. Number two, plasmolysis. Number three, hemolysis and crenation. Number two, describe the roles of diffusion in living organisms. Then number four, I said describe the roles of diffusion in living organisms. Then, and the next question, which is the last one, explain how osmosis is used in the opening and closing of the stomata. Explain how osmosis is used in the opening and closing of stomata. So those questions, we are going to look at them in our next class. I will ensure that at least each and every person will be present in our class. I will find out those who did the work and those who will not have done the work, because I know how to find out as a teacher. So I want to wish you well. Number one, the question number one was asking, define the term. I just about the last one. I've said explain how osmosis is used in the opening and closing of the stomata. The number one, I said define the following terms. One was target pressure and wall pressure. Define those two. Then number two, I said define the term plasmolysis. Number three, crenation and hemolysis. Okay, great. So thank you, and I want to wish you all the best in your classes throughout the week. And please keep safe at home. Do not make sure you are keeping following the government guidelines in relation to the COVID-19. And above all, let us keep on praying that this pandemic may come to an end someday and we can all resume our normal classes. We are actually missing our students so much. So please stay safe for us at home, and we are praying that we will soon we will soon beat the pandemic. So have a good day and may God bless all of you. Thank you. Okay.
Yeah.
Papa imeisha.